You even see my mitotic spindle, those microtubules. I know this is still metaphase, and I recognize this as anaphase, because now I can see a separation from them. I can see the remnants of the mitotic spindle. It's actually helping to push these centrosomes further apart. And again, I don't see any nuclear membrane. Over here, though, they're less condensed than these guys. They're definitely more spread out. I can't see a nuclear membrane yet, but I can see the formation of cytoplasmic membrane between these two. So it's easy to kind of tell where you are in our mitotic process. So to kind of sum up the whole thing, we go back to this earlier slide. In terms of looking at mitosis, what did we start with? We're going to start with a single cell. Now, how to think about the chromosomes. So we have 46 chromosomes, but they're in pairs, remember, one from mom, one from dad. So we can also say we have 46 separate chromosomes or 23 pairs. It's the same thing. One cell with 46 chromosomes or 23 pairs. By the time we are done with mitosis, how many cells will we have? You have 2. How many chromosomes in each of those 2 cells? 46 chromosomes. How many pairs in each of those 2 cells? 23, right? It's hopefully just an exact Xerox copy that you started off with.
So if every cell has the same DNA, how does how does my heart cell make heart cells?
That is a really cool question. So let's wind the clock back to you being a recently fertilized cell. So sperm and egg got together for a fertilization process. You have this DNA from the sperm and the DNA in the ovum. You're now going to go through your first round of mitosis. Now you have 2 cells. Then you have 4, 8, 16. By the time you get into what's known as a gastrula structure, it's like think of a basketball that's been folded into half. So you have basically a chamber that kind of looks like this. We kind of divide it where we have this big empty area here and a big empty area here. Somehow now, since all these cells that are in here came from one single cell, and we don't really quite know exactly what it is, but at some point they become what's known as pluripotent. The ability to become anything, aka a stem cell. I can take a stem cell, let's call it stem cell A, and put it in your big toe. The cells somehow know what to become based upon where they find them. I'll go to this idea. Cells have a life cycle, like a to-do list. How long their to-do list is depends upon what kind of a cell it is. Some live for hours, some live your entire life. But in that to-do list, they have checks. In other words, a moment where they pause and go, all systems go, cool, let's continue on our life cycle. And it has to get that checkpoint passed. If it doesn't get in a okay signal, it's another bunch of chemicals inside the cell itself. If it doesn't find those chemicals, then the cell simply just leads to cell death. It figures out there's something wrong somewhere, somehow, and we are now not going to pass that on to the next generation. And we go through apoptosis, which is the death of the cell. In a sense, that's what causes a lot of cancers, is that when you look at their life cycles, it's uncontrolled mitosis. They just grow and grow and grow and grow. They're not being told, hey, wait a minute, there's a boo-boo somewhere. Quit reproducing the mistake. But they do. And tumors can be huge. I remember seeing a documentary on a woman that had a 26-pound calcified tumor. And then they realized that the center of the calcified tumor was a fertilized fetus that had tried to grow outside the uterus. And it grew and grew and grew because the body had a bit of a mutation and it just started to multiply, and the body also pretended like a pearl, and it surrounded it with connective tissue to isolate it from mom, but those cells kept growing and growing, and she had this really big mass. And they realized after ultrasound that she actually had a fetus way in the center of it. But most tumors happen this way, where the cells simply don't have a stop signal, and they just keep growing and growing. Some grow big, some grow just a little teeny bit. Depends on what the tumor is. All right, here's, yeah.
Sorry.
No, it's okay, Kelly. You're cool.
Are they different from the surrounding cells, or is it like an expansion of
Well,
whatever cell is?
It's both. They're a lot like their neighboring cells, but they also take physical characteristics that are different. Like if you have a melanoma on the skin, a mole is a little bit like a tumor in a sense. Those cells had a momentary lack of communication. They just grew a little more than they're supposed to. But then they kicked in and said, oh, I guess we're supposed to stop growing. When you look at people that have these large growths on their face, it's a little bit of an excess bit of growth. Their check systems didn't work for a little bit momentarily. And then they reconnected and said, oh, I guess we need to stop growing. So they have both the similarities of their neighbors, but also a little bit different because you can actually see them and they look different. So that's why you can take a tissue sample and send it to a lab. You want to literally look at it and go, that does not look like a normal cell. Even though you can't see that it's a, quote, cancerous cell. You can look at it and go, you look a little different. Then do what's called a biopsy, and they take the DNA out, and they sequence the DNA and go, yeah, there's a mutated gene right there. And we know that that gene doesn't create any life-threatening issues, so it comes back being benign. Don't worry, it won't grow anymore, or we're going to remove it, and it may have to come back in 15, 20 years, it's going to grow again. Because we're not removing the stem cell that created in the first place.
Actually, it's something is very related to DNA and its feature something. You know, like they have any small.
All right, so how is meiosis different? First thing I want you to think about is this. When we start going through meiosis today, what time we have left, I want you to think of it, it's the same as mitosis basically. Basically, it's the same as mitosis basically. The names of the phases are the same. Right? There's going to be metaphase, anaphase, prophase, and all that stuff. But how is it different? Well, mitosis takes place in all of your cells except for ovaries and testes. Mitosis takes place everywhere except testes and ovaries. Takes place everywhere except ovaries and testes. Meiosis, yeah. So where does mitosis take place?
Everywhere else.
Everywhere but where meiosis takes place. Which is pretty easy. So if meiosis happens only happens in ovaries and testes. Then mitosis happens everywhere but ovaries and testes.
So mitosis happens everywhere but ovaries and testes.
Right.
Meiosis only happens in ovaries and testes.
Correct.
Okay.
All right, so
No.
testes.
Meiosis. No, I'm sorry. I'm really sorry. Meiosis only happens in ovaries and testes.
Exactly.
Mitosis happens everywhere but ovaries and testes.
Exactly.
Okay.
Right, that's why it's a mental mind screw. Right, meiosis, this word up here, happens in testes and ovaries, and that's it. The other process happens everywhere except
So close.
where meiosis takes place. So testes won't do mitosis. Ovaries won't do mitosis. Okay? Now, it's going to get even a little more confusing than that. Let's look at this little diagram here. And here we have a cell. Remember, we count centromeres to count chromosomes. So I have a centromere here and a centromere there. I have two chromosomes here. We're only looking at two. Even though you've got 46, we're only looking at two. What we have is that we have a chromosome from mom and a chromosome from dad, the same version of genes. That's why they're the same size. We've got a karyotype, right? The pair of the 1, the 2, the 3, the 4, the 5, right? Down to 22. There's 2 in each one of those numbers. Because one came from mom and one came from dad. They have the same code, the same genes. Well, So here's the gene for B, and here's the gene for B. They're just different versions of that gene.
Mom and dad.
Now, a different version of the gene, we call that an allele. It is a version of a gene. What do genes code for? Proteins, right? So no, it's a version of a protein. The same protein. In order to do meiosis, the first thing has to be the same as mitosis, and that is we have to go through the S phase. We have to make a copy of all of our genetic material. So this cell goes through the S phase and makes a copy of all of its chromosomes. It'll make a copy of its mom's version and a copy of the dad version. So here's the end result. I have a copy, 2 copies of mom, right, and the 2 copies of dad. And that is our classic X pattern that we see from mitosis, right? It makes just a big X. I still have 2 chromosomes because I only still have 2 centromeres. So I started with 2, but I still have 2. But now I have 2 sister chromatids for this chromosome and 2 sister chromatids for this chromosome. How many total chromatids do I have in that picture? 4. So we can refer to this as a tetrad. Because I have 4 sister chromatids. Because those 4 sister chromatids are about the same gene. Right? They're about the B gene. I've got 4 chromatids for the B gene. Now there's also another word that we're going to use to talk about these 2 chromosomes and their overall 4 sister chromatids. And that is homologous pairs. Homologous pairs. See the word pair? Right? It's a pair of chromosomes. Homologous. What does homologous mean? Means the same. We should focus in on the part called homo, which means the same. So in other words, they're the chromosomes that have the same genes. They have different versions, but it's still the same genes. Now, to set this up so that it makes sense.
Sorry.
That's okay.
In the second circle, are there 2 homologous pairs?
No, there's just one homologous pair right now. So a homologous pair is also known as a tetrad.
4.
All right, now, let's look at this really quickly. This is going to be, no, do this now. 23 in here. This is going to be an ova, a human egg cell. So this is the nucleus, this is the cytoplasm. And here is going to be what I call a DNA torpedo. And it's going to have 23 chromosomes. So when these actually come together during fertilization, if I go ova plus sperm, right, we have 23 of these plus 23 of those equals how many?
46.
That's how you get your 46 chromosomes. 23 from mom and 23 from dad. We have to use meiosis. It only happens in ovaries and testes to create an egg that only has 23 chromosomes in it, not 46. We have to create sperm cells that only have 23, not 46. Because if you took a 46 chromosome egg and fertilized with a 46 chromosome sperm, how many chromosomes do we have? 46 plus 46 is
92.
92, right? That ain't gonna work. We're not a 92 chromosome animal. So evolution, intelligent design, what you're saying says, I have a way of solving that problem. We're first going to go through DNA synthesis, make a copy of each version of chromosomes. That creates a homologous pair. I have a copy of my mom's and copies of my dad's. If this was 46 chromosomes, right, this now gives me a total of 92 chromatids. We go through the first round of meiosis. We take 92 and we're going to split the homologous pairs. Right? Moms went up here and dads went down here. We didn't separate the chromatids. We separate the homologous pairs. Now this cell only has one chromosome there, right? This one had two. This is what's known as reduction division. We're reducing the number of chromosomes. We went from 46, we doubled them to 92. First round of meiosis, we go back to 46. We're going to go through another round of meiosis, but now we separate the sister chromatids, and we're going to get two cells. One cell got one of the chromatids, the other one got the other. We do the same thing with the dad version. We're going to separate the chromatids, one cell gets one, the other cell got the other one. But we went from 46 to 23. So 2 rounds of reducing. Now what you have is 23 chromosomes. This could be an ova, it could be a sperm, and if they were fertilized together, you're back to a human 46, or 23 pairs. Okay, let's see how we do that. All right, so interphase and interphase is the same whether it's in testes or it's in your nose cell. We need to first condense the chromosomes down. So, what is the very first stage of mitosis? First stage of mitosis is called... I haven't gotten there yet, John. I'm going to peek in my notes. I'm going to say prophase. Exactly. You're going to call this prophase 1. Because we know there's 2 rounds of meiosis. I need to specify which prophase it is. Is it the first one or the second one? Here's what you do not need to call it. Don't say prophase 1 of meiosis 1. That's what the 1 implies. It's the first round of meiosis. Now, just like in mitosis, we need to condense the DNA, right, to make chromosomes. We need to move our centromeres to opposite, or centrosomes, pardon me, centrosomes to opposite sides of the cell. That's the same. And we have to get rid of the nucleus. Nothing different.